Honorable experts and dear audience, I'm sorry for the interruption. I'm continuing from where I stopped. I will be presenting Association of BDR Gene Polymorphism with Susceptibility to Ovarian Cancer in Bangladeshi women. Before starting my presentation, I want to express my sincere gratitude to my supervisor, Professor Dr. Muhammad Siddiqul Islam sir, who is present here, and Professor Dr. Abu Said MD Musaddek sir, Dr. Muhammad Wahid Akhtar sir, and Dr. Nusratun Nahar ma'am for their contributions to make it possible for me to complete the research. The background about ovarian cancer. Ovarian cancer is a major cause of death among gynecological cancers worldwide, even as well as in Bangladesh, largely due to last stage detection and lack of effective early treatment or detection, genetic susceptibility, particularly concerning the vitamin D receptor, which is acknowledged as a significant. The objective of my research was to investigate the association between BDR or vitamin D receptor gene polymorphism and the susceptibility to develop ovarian cancer in Bangladeshi women. Here are some introductory slides regarding ovarian cancer and its histological classification, detailed classification, and signs and symptoms, which is not the major concern of discussing today. And here are some key risk factors, which includes several types of mutations, age and family history, reproductive and medical history, lifestyle or environmental factors, etc. Now I'm coming to the vitamin D receptor. The vitamin D receptor belongs to a superfamily of nuclear receptors. Its gene spans of 75 kV and is located in the long arm of the chromosome. It interacts with other transcriptional factors. The best studied one is the retinoid X receptor. By binding to genes with promoters containing a vitamin D response element, the vitamin D receptor regulates gene expression. The VDR has been found in 30 different tissues. It has shown that the role of VDR stretches beyond the calcium and vitamin D metabolism. Here is a crystal structure of vitamin D, where the green marked area is the complex with calcitriol, which is yellow in color, and the position of single nucleotide polymorphism is marked with red. Genetic polymorphism, which is a variation in DNA sequence. It can be single nucleotide polymorphism, insertions, deletions, variable number of tandem repeats, and microsatellites, which is already mentioned in the previous presentation. And it is an illustration of single nucleotide polymorphism. The VDR gene polymorphism and ovarian cancer. VDR is present in normal ovarian epithelium, human ovarian tumors, and in human ovarian cancer cell lines. Overall, 83% of normal ovarian surface epithelium is VDR immunoreactive. Among polymorphisms in the vitamin D receptor gene described to date is RS2228570. Is the only single nucleotide polymorphism that has been shown to affect the VDR protein structure. This single nucleotide polymorphism is also known as foci polymorphism due to the presence or absence of the mentioned restriction enzymes site. This is the method where we have the study samples, genotyping and statistical analysis, and further studies. Our samples include 200 patients with ovarian cancer and the 200 healthy individuals. For genotyping, we have to collect the venous blood from the samples and both from the volunteers, and DNA extraction, polymerase chain reactions, and gel electrophoresis, followed by the polymerase chain reaction, restriction fragment length polymorphism for the cost effectivity, and the enzyme digestion. Further, we had to do agarose gel electrophoresis again for the confirmation. And later on, it was the statistical analysis and further studies where we have calculated or evaluated the odd ratio and the 95% confidence intervals. This is the primer sequence by which we have conducted this research, and the restriction enzyme, which is foci, and the PCR conditions by which we have conducted the PCR. This is the genomic DNA as a sample, and restriction enzymes digestion condition and the length of expected fragments, where reference or normal homozygote refers to the 341 base pair, where the mutant heterozygote is 3 fragments, 341, 196, and 145, and the mutant homozygote is 196 and 145. This is a picture of case samples, and this is the controls. Here is the PCR RFLP, and where green marked area is the primer sequence, and yellow is reverse primer, and the red is targeted SNP for which we worked on. This is the sum of the factors we have analyzed by different statistical tools, where age and some other factors were statistically significant. All of them were non-significant. That means the genetic variation or the genetic polymorphism can have a significant impact on this susceptibility to develop this disease, where you can see as a in the summary of p-values. That means the age versus TNM stage and family history, cancer, and high toxicity was significant, otherwise it was non-significant. And the result of our research include where the reference genotype is CC and the patient count for ovarian cancers and healthy individuals are mentioned here. And when it is mutated into a mutant heterozygote, that means the genotype is CT, it has a 2.41 times higher risk to develop the ovarian cancer in those certain individuals. And when it is mutant homozygote, that means both the nucleotide is mutated into alternate gene, that is the genotype is TT, where is the risk of developing ovarian cancer is 1.88 times higher. In dominant model, where the genotype is either CT or TT, it has a 2.28 times higher risk to develop the ovarian cancer in those individuals, as the p-value for all three types are statistically significant. This is the allele comparison, where T allele is significantly more risky, or it has 1.46 times higher chances to develop the ovarian cancer who is carrying it. The key findings include the VDR and this single nucleotide polymorphism gene shows a significant effect on ovarian cancer susceptibility. And the dominant model, as I already mentioned, it has the significantly higher risk, which is 2.28 times higher, and the p-value is mentioned here, and allele frequency, as I have showed. The limitations include limited general population specificity. That means it can vary due to the ethnicity or other environmental factors, which was not evaluated in this research. And the multifactorial etiology of ovarian cancer, as it is a part of several types of polymorphisms we are including on, and the vitamin D status on these people, how it can play a role in this type of polymorphism or the susceptibility. We need to assess the vitamin D level in their serums also, and we are working on it to detect the vitamin D from the serum level for both patients and healthy individuals, and some others. The future implications can be genetic susceptibility markers, and we can use it as a genetic biomarkers while we are moving to the whole genome sequencing on something like more advanced and feasible process. And early detection and prevention, personalized medicine by assessing the response and toxicities of different types of chemotherapy, which are being used in treating different types of cancers, and as a novel biomarkers. Here is all of my co-authors whose contribution made it easier to complete the whole process. Thank you to everyone, and this is a picture while showing our teammates and faculty members while producing ORS and Whitfield ointment for the flood affected people in Noakhali in 2024. Thank you everyone, thank you so much.